Hello and welcome. Today we will be exploring genomics and how genomics can be utilized by healthcare providers in their clinical practice. Genomic information is only one piece of the puzzle for why some people develop a disease and others do not. As scientists continue to discover more about how lifestyle habits, environmental factors, and genomics interact, the information can be used to provide targeted and personalized care to patients. As we navigate through this course, our objectives will include defining the terms genetics, genomics, and pharmacogenomics, identifying risks for health disorders based on information presented from pedigree charts, and improving healthcare provider confidence in using genomics in clinical practice. People often use the terms genetic and genomics together, even though they mean different things. Let us clarify the differences between the two. The definition of genetics is the study of biological inheritance and traits. Genomics is the study of the entire genome, or the complete set of genes and genetic material, of an organism. This includes the structure, composition, functions of genes and non-coding DNA, and gene-to-gene interactions related to the genome of that organism. Genomic medicine is a medical discipline that involves using a person's genomic information as part of their clinical care. Other similar terms include individualized medicine, personalized medicine, and precision medicine. Additionally, precision medicine or precision healthcare is medical care that takes advantage of large data sets of individuals, such as their genome, to tailor their healthcare to their unique attributes. No two individuals are the same, and so their care should be individualized based on their specific genes and associated risks. The Human Genome Project started in the mid-1980s. In May of 1986, the Department of Energy Office of Health and Environmental Research held the Genomics Sequencing Workshop in Santa Fe, New Mexico, to explore the feasibility of pursuing a human genome project. In 1988, the National Institute of Health lays out a plan for the project and establishes an advisory committee to advise the NIH on research related to genomic analysis. A 5-year plan with goals for the project is published in 1990, and the National Advisory Council for Human Genome Research is established. It isn't until 1996 that human DNA sequencing begins with pilot studies at 6 universities in the United States. That same year, the Center for Inherited Disease Research is established at the Johns Hopkins Bayview Medical Center. The first gene associated with Parkinson's disease and the first major gene that predisposes men to prostate cancer were identified. It wasn't until 1999 that large-scale sequencing of DNA began. 85% of the human genome had been sequenced by 2000. In April 2003, the Human Genome Project was completed. The United States Senate passed the Genetic Information Non-Discrimination, or GINA, Act in 2008 that prevents health insurers and employers from using genetic information to determine eligibility, set premiums, or to hire and fire people. In 2010, the Genetics and Genomics Competency Center, G2C2, launches an online tool to help educators teach nurses and providers about genomics. The NIH developed guidelines for polygenic risk scores in 2021, and in 2022 the first complete and gapless sequence of a human genome was complete. With the exception of mature red blood cells and cornified cells, think hair, skin, and nail cells, all cells in the human body contain a nucleus which contain the human genome. Humans have 23 pairs of chromosomes packed into the nucleus of each cell. Each chromosome is rod-shaped and packed with long stretches of DNA. The structure of chromosomes keeps DNA wound around proteins called histones. Without the histones, DNA molecules would be too long to fit inside the cells. If all of the DNA molecules in a single human cell were unwound from their histones and placed end to end, they would stretch to be about 6 feet. This karyotype demonstrates an example of a chromosome map for a human. There are 22 pairs of autosomes and 1 pair of sex chromosomes. The first pair of autosomes is labeled number 1 and is the longest, while the 22nd pair is the shortest. The sex chromosomes are different sizes. The X chromosome, which females have 2 copies of, is about 3 times larger than the Y chromosome. It contains approximately 900 genes, compared to the Y chromosome that has about 55 genes. In the karyotype above, there is an example for both a male and a female. The male pair, XY, is on the left and the female pair, XX, is on the right. Deoxyribonucleic acid, or DNA, is a double-stranded macromolecule that is made up of a sugar and phosphate backbone coupled to sequence bases or nucleotides. The nucleotides that make up base pairs in gene coding include adenine, cytosine, guanine, and thymine. These nucleotides are paired up in pairs to make bases. Each person's DNA is made up of nearly 6 billion nucleotides, or 3 billion base pairs. Genes are groups of nucleotides and segments of DNA which carry specific training or instructions that are necessary for cells to perform their given function. Genes are the basic unit of inheritance, are passed down from parent to child. They contain information to specify physical and biological traits. The human genome has about 25,000 genes in total. Each cell in the human body contains a complete set of genes for that individual. DNA from each cell nucleus is used to make proteins in a 2-step process. The first step is referred to as transcription. During this step, enzymes read the information in the DNA molecule and transcribe it into messenger ribonucleic acid, or messenger RNA, which acts as an intermediary molecule. Then, the messenger RNA molecule is translated into amino acids, which are the building block of proteins. The messenger RNA communicates the order that amino acids should be produced so that specific proteins are made. Amino acids can be produced in many different combinations to form a large variety of proteins and genes. Next, we will look at the differences between a genotype and a phenotype. A genotype is the specific alleles that a person has for a gene or locus. It is considered to be the genetic blueprint. Dominant alleles are represented by capital letters, and recessive alleles are demonstrated by lowercase letters. A patient can be homozygous, meaning they carry 2 of the same alleles for a gene, or heterozygous, where they carry 1 dominant and 1 recessive allele. A person's phenotype is the physical presentation of their genes. You can physically see if someone has blonde hair or blue eyes. A person with a heterozygous or homozygous dominant gene will likely physically look the same in the example that is demonstrated by the pea plants. Although they appear to be the same, a heterozygous and homozygous dominant gene will not have the same genotype. One example of how an environment can affect a phenotype is in flamingos. Flamingos all have the same genotype for their pink appearance, but only birds who have a diet rich in carotenoid pigments have colorful feathers. Birds with bright pink feathers who have their diet changed from a high carotenoid pigment content to lower content will fade in color. Another human example includes identical twins. Identical twins share the same genotype, yet they often have different phenotypes, height, weight, and fingerprints. This is because external factors such as diet and exercise affect gene expression. Genomic variations can occur on a varying degree from a single DNA nucleotide change to deletions or duplications of an entire chromosome. All people have the same set of genes, BRCA for example, and 99% of DNA is similar between all humans. sequence variants that change one nucleotide in the DNA sequence, and that differ between individuals or even humans and other species are known as single nucleotide polymorphisms, or SNPs, and occur quite frequently across the human genome. SNPs are currently being studied extensively in association with disease and drug response. DNA alterations cause disease by altering protein production. Sometimes it is assumed that people who have the same diagnosis or disorder have the same mutation. However, it is important to note that people with the same genetic condition likely have unique or different mutations. This is referred to as genetic heterogeneity. Alzheimer's disease and cystic fibrosis are two examples of diseases that are caused by a variety of genetic mutations. Germline mutations are mutations that originate in a person's gamete or reproductive egg or sperm cells and affect all cells in the growing organism. This means that every cell in the human body will have the mutation present for that individual. Half of that individual's future sperm or eggs will also carry the mutation, which could affect the offspring for future generations. A somatic mutation is a mutation that occurs due to damage of a single human cell sometime during the individual's lifetime, and is absent in the individual's original germ cells. A somatic mutation is one of the most common causes of cancers. These cancers are referred to as sporadic cancers. Only tissue derived from the mutated cell will be affected. None of the individual's sperm or eggs will carry the mutation, meaning the mutation will not be passed on to offspring. Although most diseases and physical characteristics in humans are polygenic, meaning they are determined by more than one gene, we will demonstrate dominance through discussion of disorders that are caused by single gene mutations. Phenotypes due to alterations at a single gene are referred to as Mendelian or monogenic diseases. These disorders exhibit significant effects on gene mutations and are named after Gregor Mendel, the biologist who studied the reproducibility and recurrence of variation in garden peas. Examples of Mendelian disorders are cystic fibrosis, sickle cell, and hemophilia. Monogenic human characteristics include the ability to roll your tongue, whether or not you have attached ear lobes, and the presence of a widow's peak at your hairline. Over 80% of Mendelian disorders known to date have a pediatric onset phenotype. If you look at the Punnett square, you will see where one parent's genotype is listed at the top and the other parent is listed on the left side. Both parents, for this example, are heterozygous for the gene that determines whether or not a person can roll their tongue. The ability to do this is a dominant gene, so let's determine the likelihood that the offspring will also be able to roll their tongue. Next, we will take the alleles for the parent on the left side of the table and distribute each allele into a separate row. The resulting pairs demonstrate the potential allele combinations from each parent. The top left box demonstrates two capital X alleles, which indicates that there is a 25% chance that an offspring would be homozygous for the dominant allele, allowing the offspring to be able to roll the tongue. 50% of the offspring would be heterozygous, but would still be able to roll their tongues. The 75% of the offspring represented by these two groups would also have the same phenotype, or the ability to roll their tongue, despite having different genotypes. Finally, there would be a 25% likelihood that an offspring would end up with homozygous recessive alleles, which would prevent the offspring from having the ability to roll the tongue. Now that we have refreshed the concept of dominance, and what it means for gene expression in patients, we will continue to discuss the different types of inheritance and what they may look like in a pedigree chart. Autosomal disorders are those disorders that occur in the autosomes. The sex chromosomes are not affected, and those inheritance patterns will be discussed later. Autosomal disorders are inherited equally by males and females, and will include both dominant and recessive disorders. Autosomal dominance is a pattern of inheritance in which an affected individual has one copy of a mutant gene and one normal gene, or two copies of a mutant gene. The frequency of sporadic cases is positively associated with the severity of the phenotype. Autosomal dominant phenotypes are often age-dependent, less severe than autosomal recessive phenotypes, and associated with malformations or other physical features. One example of an autosomal dominant disorder is Huntington's disease. The example that is shown includes a father who is heterozygous for the mutant gene and a mother who is unaffected, and each offspring has a 50% chance of being affected. Autosomal dominant disorders are significant because mutations on a single allele are sufficient to cause disease. Here is an example of a pedigree that demonstrates an autosomal dominant inheritance pattern. When looking at this in the clinical setting, there are several observations that can be made. First, remember that each offspring will have a 50% chance of being affected. This does not mean that if parents have two children, only one will be affected. Instead, remember that each offspring will have a 50% risk for inheriting the mutation. Risk of inheritance is fixed and will be independent of the number of offspring. Additionally, males and females will be affected equally. When assessing a family pedigree, a vertical pattern is observed and multiple generations are affected. Autosomal recessive disorders occur when an affected individual has two copies of a mutant gene on a pair of chromosomes. When a patient or individual has an autosomal recessive disorder, the parents are known to be obligate carriers. In these individuals, mutated alleles results in a partial or complete loss of function. Examples of autosomal recessive diseases include sickle cell anemia, cystic fibrosis, and thalassemia. Heterozygous carriers of a defective allele, which could include obligate carriers, may display subtle differences in phenotype that become apparent only after precise testing or in the content of environmental influences. In this pedigree, the parents are heterozygous for the recessive gene, making them obligate carriers. In this situation, there is a 25% chance that offspring will be affected, a 50% chance that offspring will be a heterozygous carrier, and a 25% chance that offspring will not have any mutated alleles. Pedigrees for autosomal recessive disorders often demonstrate a horizontal pattern, rather than vertical. Furthermore, a significant portion of a single generation may be affected. It is important to note that in plain pedigree charts, it is easy to miss individuals who may be carriers if only affected and unaffected notations are used. Instead, if a recessive disorder is known to be present, it would be helpful to add in notation or a more detailed version to note known carriers. Males and females are affected with equal frequency and severity. When both parents are affected, 100% of the offspring will be affected. Affected individuals who mate with unaffected individuals who are not carriers will only have offspring who are carriers. Additionally, the rarer the recessive phenotype, the more likely it is that the parents are consanguineous or are related. This is sometimes seen in royal families in Europe. X-linked disorders are those that are related to the sex chromosome pair. These disorders may be dominant or recessive. X-linked dominant disorders are often lethal or very severe in males, and appear predominantly in affected females. They are also often associated with reduced reproductive fitness due to the severity of their phenotypes. Because males only have one X chromosome, they are referred to as being homozygous for the mutant allele. Males are also more likely to develop the mutant phenotype, regardless of whether the mutation is dominant or recessive, because there is only one X chromosome. A daughter will always inherit her father's X chromosome in addition to one of her mother's two, and will either be heterozygous or homozygous for the mutant allele. The risk of developing a disease due to a mutant X chromosome gene differs in the two sexes. Characteristics of X-linked dominant disorders include that females are affected more than males. Both males and females of the same generation can be affected if the mother is the affected parent. heterozygous women transmit the mutant gene to 50% of their sons, who are affected, and to 50% of their daughters, who will be heterozygous if their father is unaffected. If an affected male mates with a heterozygous female, 50% of the male offspring will be affected, giving the false impression of male-to-male transmission. Among the female offspring of the same mating, 50% will be affected as severely as the average homozygous male. In small pedigree charts, this may mimic the appearance of an autosomal dominant inheritance pattern. X-linked recessive disorders rarely produce symptoms in females, as females typically have an unaltered copy for their second X chromosome. When a female is a carrier for an X-linked recessive condition, each daughter has a 50% chance of also being a carrier. Each son will have a 50% chance of inheriting the mutant gene and having the condition, because they only have one copy of the X chromosome. One unique point for X-linked recessive disorders is that females may manifest features of the mutation even if they do not have homozygous alleles. Normally, one of the two X chromosomes is shut down in each cell to avoid excessive X chromosome genes being expressed. It is normally random in each person which X chromosome is inactivated, whether it is from the mother or father, but there is typically around 50% expression from each X chromosome. Sometimes the inactivation can be skewed, leading to a decreased expression of the normal allele and manifestation of features of the disease from the mutated X chromosome. Examples of X-linked recessive disorders include hemophilia A, Fragile X syndrome, and Duchenne muscular dystrophy. There are fewer characteristics for X-linked recessive disorders than for other inheritance patterns. X-linked recessive disorders cannot be passed from father to sons. However, females can become carriers from affected fathers. Males are almost exclusively affected, as females who are carriers may pass the recessive gene to their sons. When looking at a pedigree, females may be marked as unaffected when they may truly be carriers for the disease. This will typically happen if genetic testing has not been completed. Mitochondrial inheritance patterns are determined by the mother's genes, as sperm cells do not contribute mitochondria during conception. A man who is affected will not pass it on to his offspring, but an affected mother will pass it to all of her offspring. Because this inheritance pattern is straightforward, only pedigrees will be presented. On the left you can see the pattern for an affected female, and on the right is the pattern for the affected male. Mitochondrial diseases include Leig's disease and mitochondrial encephalomyopathy, lactic acidosis, and stroke-like syndrome, or MELAS. Mitochondrial DNA is what is used to compare DNA of ancient populations in archaeology to determine patterns of ancient migration and population habits. Multifactorial inheritance is when a disorder is caused by a combination of genetic and environmental causes. Examples include cleft palate, heart disease, and diabetes. Gene-environment interaction between genes and the physical and social environment, and as mentioned earlier, has an effect on phenotypes. Most human traits and diseases are influenced by how one or more genes interact in complex ways with environmental factors such as chemicals in the air and water, nutrition, and ultraviolet radiation from the sun. The impact of an environmental exposure on disease risk is different for people with different genotypes. Additionally, the impact of a genotype on disease risk is different in people with different environmental exposures. One example of a gene-environment interaction involves the NAT2 gene, the environmental factor of smoking, and bladder cancer. Tobacco smoking is a known risk factor for bladder cancer. According to the American Cancer Society, smokers are at least 3 times as likely to get bladder cancer as non-smokers. Smokers with one variant in NAT2 have a much higher risk of bladder cancer compared to smokers with a different variant. This is one example of how both a genetic variation and an environmental factor play a role in disease risk. Family histories have traditionally been used in the clinical setting to identify health risks for each patient. Current evidence has demonstrated that collection and analysis of a comprehensive family history supports use of genomics in clinical practice and can be utilized to prioritize who should receive more expensive genomic or diagnostic testing. When collecting a family history, it is important to gather at least 3 generations' worth of information so that patterns of inheritance are established. Once the information is collected, the information can be assessed to determine which patients have higher risk for genetic factors that could lead to disease development. Identification of risks or red flags early allows for prevention and early treatment of some diseases, which helps improve patient outcomes and potentially decreases overall healthcare costs. Pharmacogenomics is the study of how an individual's genetic attributes affects the body's response to medications and demonstrates how genomics can be applied to patient care. Goals in the clinical setting include selecting the right medication for the right person at the right dose. Individuals who have gene mutations may have an unpredictable response to a medication. Medications work on receptors in the body, which are made up of proteins. It is estimated that only 25 to 60% of patients respond appropriately to their medication, and that the rest either do not receive an appropriate medication or experience some degree of adverse drug reactions. Additionally, it is estimated that over 20% of prescription medications in the United States have pharmacogenomics recommendations based on known genetic variants. Appropriate use of pharmacogenomics can help with development of newer and safer medications, improve patient engagement and compliance with treatment, improve medication selection, and improve patient safety. Ultimately, integration of genomics and pharmacogenomics into clinical practice can help decrease healthcare costs and improve patient outcomes. Direct-to-consumer genetic testing includes genetic tests that are sold directly to consumers to provide information about their genetic information. These types of tests are gaining popularity, and companies like 23andMe and Ancestry.com are just 2 of the many companies that advertise their genetic testing to help identify ancestry, health traits, and health risks. Usually, these tests are completed using a saliva sample, and healthcare providers are not involved in discussing results with patients. A large concern with these types of tests include the potential for information to be misunderstood by customers who have had no training with genetics or genomics. Clinic-based genetic testing and screening for clinical purposes can be ordered by any healthcare provider. These tests are ordered and completed at a clinic, and results are communicated to patients by providers who have had training and education in genomics. If genetic screening is ordered and there is a positive result, the patient has a confirmed gene change or genetic mutation in one or more of the genes that were analyzed. A patient who has a positive genetic screen is at a higher risk for developing a hereditary condition. Negative genetic screen results means that no gene mutations were identified. Although this is typically a good finding, it does not specifically rule out a genetic condition because there may be undiscovered genes or variants that were not included in the testing. Occasionally, a genetic test result will come back with a label of uncertain. This means that at least one variant was found, but it is uncertain what those variants could be contributing to a disease or disease risk. Uncertain results are relatively common when a large number of genes are analyzed. Finally, there are several reasons why individuals may ask to have carrier testing completed. One of the most common examples is that a couple who is considering having children may choose to have carrier testing done if one of the individuals is a known carrier for a recessive disorder. This allows the couple to be able to calculate the risk that their offspring would have for inheriting a genetic condition, and plan ahead for those calculated risks. Thank you for completing this genomics presentation. Have a great day.